Hello, welcome to our lesson about antimicrobials that target cell walls. Most of them are known as beta-lactams. Alright, this lesson is going to go over how bacteria make their cell wall and how beta-lactams can be used to inhibit bacterial cell wall synthesis. Okay, we're also going to go over beta-lactamases and beta-lactamase inhibitors, and we're also going to go over a special drug known as vancomycin, which does not really fit into the beta-lactam family. Now first you need to know how bacteria make their cell wall and how they keep up integrity. What you need to know is that bacterial cell wall are made out of peptidoglycans. Now it's just exactly how it sounds. It's got a glycan in its name, so you want to think of it a sugar group, and a peptido in its name, in other words, an amino group. So these polysaccharide backbones also have these alternating NAM and NAG. So you could see over here, these polysaccharides over here have alternating NAM and NAG. And each NAM actually have an alanine group. So if each NAM have an alanine group and they're right there adjacent to each other, two alanines can make a peptide bond. So here's the peptide bond, and there are two alanine groups on either side. So this peptide bond is made by an important enzyme, and this enzyme is known as transpeptidase. Transpeptidase also has another name for it. It's also known as penicillin binding protein because it is the target of beta-lactams such as penicillin. But the important thing that you need to know is that this transpeptidase makes the peptide bonds between the adjacent alanine groups. This peptide bond is going to be called D-ala-D-ala linkage. And this D-ala-D-ala linkage adds integrity to the bacterial cell walls, and it prevents it from bursting. Now beta-lactams are going to prevent the formation of D-ala-D-ala linkage, and they work by inhibiting the transpeptidases or the penicillin binding protein. So whenever I say beta-lactam, I want you to automatically think of the penicillin group, the penicillin family. And they're going to target the penicillin binding protein, which is the transpeptidase. There's a lot of different names for each of these stuff. So if you inhibit the transpeptidase, what does that mean? So over here I'm showing you some beta-lactams. Normally what you have is just a transpeptidase that goes in and makes a D-ala linkage, as we discussed before. But if you have these beta-lactamases, they mimic the D-ala-D-ala linkage, and they go in here and they pretty much make the transpeptidase useless. So that pretty much ruins the integrity of the cell wall and makes the bacteria more prone to burst. And over here I showed you a picture of penicillin, which is a type of beta-lactam, and here's the beta-lactam ring. It mimics this peptide bond that you see with D-ala-D-ala, but not quite enough. It's just enough to trick the transpeptidase. And this is bactericidal because the bacteria will actually burst and die. It doesn't just inhibit the reproduction of the bacteria, it actually causes them to burst. Now you can develop resistance to these beta-lactams, and the resistance is built through beta-lactamase inhibitors, which we will talk about later. But these beta-lactamase inhibitors, just as an overview, they just basically cleave the beta-lactam ring, and if you break that beta-lactam ring, the penicillin pretty much becomes useless because it can no longer trick the transpeptidase. And this is just a way for bacteria to develop resistance towards beta-lactam. Now, we're going to talk a lot about spectrum, and that's more of a third year, fourth year kind of thing, but it's important for you to understand which organisms are easy and which organisms are difficult to treat. I just put them in pretty much the order of treatment. Usually Staph aureus is affected by a lot of our antibiotics, and that's good because a lot of infections are actually caused by Staph aureus. Then you've just got gram-positives in general. They tend to be more sensitive. Now gram-negatives are a little bit harder, and I'll tell you why. If you look at a gram-positive, what they have is they have their cell wall exposed to the environment. So whenever we add antibiotics, they can damage the cell wall immediately. But a gram-negative, while the cell wall is smaller, they also have these external membranes, and these external membranes can have pore-in channels that are selective, and they can pretty much inhibit our drugs from going in. So our drugs have a difficult time reaching their cell wall. It limits the amount of drugs that we can use on them. So that's why gram-negatives tend to be a bit harder than the gram-positives, and it requires more coverage. Now, one of the gram-negative species, Pseudomonas, is particularly difficult. It is a hospital-acquired disease, and it requires specific drugs. It's just developed mechanisms of resistance to a lot of our drugs, and it can cause a lot of very deadly diseases, which I'm sure you'll learn throughout your module. Another one that we also find in the hospital is MRSA. MRSA just pretty much means methicillin-resistant Staph aureus, and that just means that it's a Staph aureus type that is resistant to our normal beta-lactam, and so we have to come up with new drugs to target it. Okay, penicillin, the original beta-lactam. You've probably already heard the story about the researcher who left his agar out and some fungus started growing in it, and then he noticed the bacteria just wasn't growing on the agar anymore. And that's how you really discovered penicillin. It comes from a species of fungi. So all you really need to know is that it's a beta-lactam. It has the same mechanism, inhibits transpeptidase or penicillin binding protein, and has the same resistance through beta-lactamase. Beta-lactamase is produced by the bacteria. Now, as far as spectrum, this is more of a third year stuff, but as far as a first and second year, what you need to know is that it treats a certain types of diseases. I've listed 3 of them. These 3 diseases are going to be the strep pyogenes. If you see strep pyogenes, it is very, very important that you give them penicillin early. And that's because penicillin will prevent rheumatic fever. So rheumatic fever, you'll learn that later. It's the Jones criteria, and you'll learn that as you go through medical school. But the important thing is that if you see strep pyogenes, you give it penicillin to prevent rheumatic fever. Another one that you'll see is going to be actinomyces. Actinomyces is usually infection around the jaw area, and it has sulfur granules in there. And so what you want to know is that you want to treat this. If you see actinomyces, it tends to be sensitive. Finally, you want to treat it T. pallidum, that is going to be syphilis. And this is one of those diseases that have just never really developed resistance towards penicillin up until now. We don't know why, but we're very lucky that syphilis responds very well to penicillin. Now, an adverse reaction, of course, is going to be a rash. Most people won't ask you about this rash because a rash can really occur with any drugs that you're sensitive to. But a more specific reaction, a more specific immunological reaction that is found in penicillin is a Coombs positive hemolytic anemia. And what Coombs positive means is that the antibiotics are bound directly onto the red blood cells. So when I say Coombs positive, I just mean that the antibodies have bound directly onto the red blood cells and it's causing a hemolytic, or it's breaking down the red blood cell anemia, which means you have low red blood cells. That's all it is. Sometimes it'll bring up penicillin in the case of the use of probenecid. I'm sure you guys have learned that with the gout treatment. Now, probenecid can decrease the renal excretion of penicillin, and it can make penicillin last longer. Historically speaking, probenecid was used in war because a lot of the people that are in trenches often got infections. So you often gave them 1 dose of penicillin and then probenecid, and then it would increase the half-life of penicillin inside of your bloodstream, so it can last a lot longer. Alright, aminopenicillins, they're pretty easy to recognize because they all start with AM, like amoxicillin and ampicillin are the 2 most common types. And they're just beta-lactams, just like penicillin. They have the beta-lactam ring that you find in the penicillin. But their R group is different in that it is more of an amino group. So over here you see a tyrosine group, or you could see a phenylalanine group, whichever one. But the whole point of it is that we're trying to trick the bacteria into thinking that this is food. And this can be really important for penetrating the porin channels. As I told you earlier, gram-negatives tend to be more difficult to treat than gram-positive because they have an outer membrane with selective porin channels. If we can trick these channels into thinking that these are amino acids rather than antibiotics, we can possibly get in there and damage their cell wall and cause them to burst. Again, the adverse effect is just a rash, but most people don't really ask that. Now, what they do ask is pseudomembranous colitis. You got to think of this as a more broad spectrum antibiotic. If you are going to be broader spectrum, you're going to target a lot of gram-negative bacteria. And these gram-negative bacteria may be bad and you might want to get rid of them, but it's not just going to affect those ones. It's also going to affect the good ones in your gut. So you're going to kill all the good ones in your gut. And that puts you at risk of some bacteria, more importantly C. difficile, to colonize your gut. And when it colonizes it, it can cause an inflammation and from all your immune system trying to fight it off. And this inflammation will cause pseudomembranous colitis. So if you wanted to review that, you could just go look up C. difficile and see how that mechanism works. Now, resistance is just like any beta-lactam. You can make a beta-lactamase, which is just to break the ring. So the bacteria can make a beta-lactamase and just break it out. What we can do to fight off this beta-lactamase is add a beta-lactamase inhibitor. And these beta-lactamase inhibitors like clavulanic and tazobactam, anything that ends with bactam really, can often prevent the breakdown. So we can pretty much prevent the resistance. So let's talk about beta-lactamase inhibitors. We already know what a beta-lactam is. It's just anything with this ring over here, like penicillin or amoxicillin. And we already know what a beta-lactamase does. It just breaks down that ring. Here I've showed you a beta-lactamase, and you could see it over here. It just breaks down the ring, and now this antibiotic is pretty much made useless. Now what we can do is we can add a beta-lactamase inhibitor, and I've showed you over here a beta-lactamase inhibitor. It looks like a beta-lactam ring, I suppose, but what it's really intended to do is it's intended to go into the active site of the beta-lactamase and pretty much make this useless. So our antibiotic can stay fresh and useful, and we can have a strong effect on bacteria that would otherwise be resistant. Now, the beta-lactamases that you need to know are going to be clavulanic acid, which was one of the first ones, or anything really that ends with a bactam. So bactam or beta-lactamase inhibitor. And I've already explained to you the mechanism right over here. Now, what you need to know is for memory, the way I like to remember it is bactam just kind of sounds like beta-lactamase inhibitor. It just kind of like seems like you just kind of crushed it all together. And clavulanate kind of sounds like cleaving, and it just prevents the cleavage of the beta-lactams. Penicillinase-resistant penicillins. So sometimes we can make penicillins that are just naturally resistant to penicillinases. And how we do that is we just add a large R group. So as you can see over here, the penicillin has just this beta-lactam ring, and so does oxacillin. Oxacillin being the penicillinase resistant one. Now, the only real difference is that the R group is a huge, bulky R group. It's just too big for the beta-lactamases to wrap around. And because of that, it gives them an advantage. They really just can't reach the beta-lactam ring. Now, the ones that you're looking for, the ones that you need to know is going to be NAF. Sometimes they learn that as NAF for staph because pretty much all of these really only used for Antistaphylococcal. So we learn it as NAF for staph. Or decloxacillin, oxa, or just oxacillin. And I remember this as just as big as an ox. So you can learn it as NAF for staph, or R group as big as an ox. And if you remember that this is a large R group. Now, what you want to know is that while this does give you natural resistance to the beta-lactamases, it does not seem to work on gram-negatives anymore. So it's just kind of a trade-off because the R groups are just so big, they just will not fit into the gram-negative walls. They're only going to really work on the gram-positives such as Staph aureus. Now, there is an exception to that. The only time it doesn't work on Staph aureus is if it is an MRSA. Methicillin being the prototype for these penicillinase-resistant penicillins. So methicillin-resistant Staph aureus is often a hospital-acquired infection that you kind of have to think of a different antibiotic. All right, antipseudomonal penicillins. We've already talked about Pseudomonas and how it can develop resistance to a lot of drugs and how it can cause a lot of diseases. A lot of the times when the Pseudomonas, they'll describe it as green tinted on agar. You can see just right here, you see that green tint. Now, you only really need to know 3 drugs in the penicillin group that is effective against Pseudomonas, and that's going to be ticicillin, carbapenems, and piperacillin. And the way I learned that is take care of Pseudomonas. As long as you remember take care of Pseudomonas, you'll remember ticicillin, carbapenems, and piperacillin. And these 3 drugs are not just only good for gram-negatives, but again, remember Pseudomonas. They don't really ask you very often on your step 1 about whether this antibiotic will work for this bacteria, but for Pseudomonas it tends to be the exception. Now, cephalosporin is a large group, and they have different generations, and you have to learn which one of the cephalosporins are in which generation. They're pretty much beta-lactams that are already less susceptible to beta-lactamases. So you don't have to add clavulanate to them. And they work really, really well when you go higher up in the generations. You don't need to know all of them, just the major ones. And I've underlined some of the major ones and what you need to know about them. So the important ones, just knowing the generation that they're in can help you out a lot. Like cefzolin and lex. I just remember that as Lin drives Alexis. Now, if you remember that, and then you remember the PEX mnemonic, which you'll find in your first aid, and all the gram-positive, you'll know the coverage. The coverage is not often asked, so you don't really need to memorize the coverage, except for very specific ones, which we'll go over. Another one that you need to know about the second generation cephalosporins are going to be fake fox fur. And I just go by whatever sounds like that, the fake fox fur. So fake fox fur. I know that if it has a ceph in its name and it has the fake fox fur, I know it's in the second generation. And if I really wanted to know the coverage for third year, I can use the Henspec mnemonic, which is just grams positive plus the Henspec mnemonic, H. influenza, Enterobacter, Neisseria, Serratia, and then the previous PEX mnemonic being Proteus, E. coli, and Klebsiella. Now here's where we get to the important ones, okay? Ceftriaxone. Ceftriaxone, you need to know what it treats. Ceftriaxone is used for gonorrhea. You need to know that. It's used for Lyme disease. You need to know that. And it's used for meningitis. It has good central nervous system penetration, and you need to know that, okay? Another one you need to know is going to be ceftazidime. Ceftazidime, I think of the mnemonic ceftaz for Pseudomonas. Ceftazidime is the only one that really covers Pseudomonas in the third generation. So it's just kind of an exception to the third generation. It's a little bit broader coverage. So the way you remember it is ceftaz for Pseudomonas. Another one that you could also remember is going to be cefepime. Cefepime is also used for Pseudomonas. Or finally the fifth generation, which is going to be ceftaroline, and that covers MRSA. Right? But the most important ones, the most commonly asked, is going to be ceftriaxone, and then occasionally ceftazidime. Now, as for the adverse effects, you do need to know, of course, you can be allergic to it. You can have an autoimmune hemolytic allergy, just like any of the other penicillins. But more specifically, it can also kill gut bacteria. And we've talked about some of the bad effects of killing gut bacteria, like pseudomembranous colitis. But you do have to also look out for vitamin K deficiency. You kill the bacteria that is making the vitamin K for you for free. Another reaction that you also need to look out for is the disulfiram reaction. Disulfiram is just a drug that we use to give for alcoholics, and it pretty much makes you have a hangover if you drink alcohol. And pretty much if you take these drugs and you drink alcohol, you will have a really bad time. Another thing is going to be it increases nephrotoxicity. So it increases nephrotoxicity, especially when they're put in with aminoglycosides, which we'll talk about in a different module of antibiotics. Moving on to carbapenems, these are the ones that we tend to reserve for just the end case scenarios. Carbapenems almost always end with penems, and you think of it as penicillins. They do have pen in their name because they are kind of beta-lactams. They still have that beta-lactam ring. But they are pretty much very resistant to beta-lactamases. So they're very resistant. And we pretty much reserve them for the superbugs, the life-threatening ones. And I'll tell you why in a second. Something that they do tend to ask is going to be, how do you increase their half-life? We don't want to keep them. We don't want to let them build up too high in our body because of all the toxicities that they may have. So we want to keep them at a low level, but we want to keep that low level for as long as possible. So we use celastin. Celastin keeps it lasting longer. Now, celastin is just inhibiting the breakdown of carbapenems inside of your kidneys. That's all you need to know. Some people think that it's going to inhibit the excretion from your kidneys. That's not true. It inhibits the breakdown in your kidneys. So what you can do is you can put them on a lower dose and it lasts a lot longer. And this saves you from the adverse effects that you get from high doses, like GI distress, very high incidence of rash. But the most important, the most dangerous one is going to be central nervous system toxicity. It gives you seizures, and that's why we don't like to use it very often. Aztreonam is a very specific beta-lactam. You can see over here it has a beta-lactam ring, so it does still fall under that family, but it falls under the subfamily of monobactam. It's important for you to know that it is not only resistant to beta-lactamases, but it actually inhibits beta-lactamases, so that makes it really good. The most important thing that you need to know is that Aztreonam is used for Pseudomonas, and I remember that as Pseudomonam. And it only works for Pseudomonas because it really only works on the transpeptidases or penicillin binding protein type 3. And this is only found in aerobic gram-negatives. Most gram-negatives are actually anaerobic. And the one aerobic gram-negative that we are really interested in medically is pretty much Pseudomonas. Now, it is used for penicillin allergies. So if you don't want to use the Pseudomonas specific beta-lactams, you could use this, and it has a much lower rate of allergies than a lot of the other penicillins. And it's also used for renal failure. It often is excreted by the liver. It doesn't really have any adverse effects, so it's very unique, but it's very narrow spectrum, only works on Pseudomonas. Finally, let's talk about vancomycin. It's very unique compared to the beta-lactams, although it kind of has a similar mechanism. As you can see, it doesn't really have a beta-lactam ring. It's just a huge group right there. And what it does is, instead of binding onto the transpeptidases, it binds onto the D-ala-D-ala linkage itself. And what it does is it makes this huge bulky group that prevents transpeptidases from latching on around that area. And the only real resistance to it is found in enterococcuses. We call these enterococcuses the vancomycin resistance enterococcuses. And the only way you can develop resistance towards vancomycin is to not have the D-ala-D-ala linkage, because that's what it binds to. Some bacteria in the Enterococcus species have a D-ala-D-lac linkage, and this is often asked in step 1 or NBME style questions, because they want you to know that the only way you can develop resistance to vanc is to have a D-ala-D-lac linkage. Indications for it is going to be pretty much all gram-positives. Even MRSA is sensitive to it. Even the ones that are very difficult to treat, like C. difficile, the enterococcus, given that it's not resistant, and the epidermidis, which often infects the valves inside of some patients' hearts. Something that you do need to know for GI, and you'll go over it again, is that it has zero bioavailability. So what that means is that if you give it orally, it'll only treat the bugs inside of your GI. It won't go systemically. If you want to treat something that is outside of your GI, you have to give it IV, and that's going to be pretty problematic, as you'll see pretty soon. Problems with IV, thrombophlebitis. That just means that you get inflammation of your veins, and it just causes a lot of inflammation. It's just a huge bulky group. Your body cannot handle it, and your body often reacts negatively to it, and it can cause this itchy inflammation all the way up your veins. Another thing is that it can cause ototoxicity, just problems with your ears, hearing, ringing of the ears. But one of the very unique side effects to vancomycin is going to be red man syndrome. That's going to be with very fast IV infusion. And this red man syndrome is the mast cell degranulation in the head, neck, and upper torso after you have infused them with vancomycin at a very rapid pace. And so the treatment for this is just to give them some anti-inflammatory stuff like Benadryl and start infusing them slower. Just slow down the infusion. And finally, nephrotoxicity. It can cause damage to your kidneys. How are you going to use this information? Don't get overwhelmed, okay? You don't necessarily need to know the coverage except for the more specific ones like Pseudomonas or MRSA. Maybe you need to learn about the coverage of the niche ones, like penicillin is good for syphilis. Other than that, focus on the mechanism, which should be pretty easy because they share the same mechanism, and focus on the unique deadly adverse effects. You need to understand how They are used to inhibit the transpeptidases and how the bacteria can develop resistance by making a beta-lactamase. And so we can fight off the beta-lactamases, and then the bacteria can develop a resistance by changing its d-ala-d-lac. So just understand that this is kind of a tug-of-war kind of game that we're playing with the bacteria and how they can develop resistance and how we can combat that resistance. I've given you a question here. You're a third-year medical student. You go into inpatient and you see that he's dying from sepsis. You have no idea what the bacteria is, and the culture in lab takes days to come out. You need to cover the big ones. So you're going to give vancomycin. You need to put in vancomycin as quickly as possible because you want to cover MRSA. You also want to give something to cover the gram-negatives as well because you just don't know what it is. So you're going to add something like ceftazidime, cefepime, maybe something like ceftriaxone, piperacillin, so you can make sure that you're getting Pseudomonas if that's what you're worried about, or Aztreonam if you're pretty much sure that it is Pseudomonas. So a lot of the times when you don't know what the infection is, you want to kind of get both, a little bit from both, the gram-positive coverage and the gram-negative coverage. That's pretty much all, and that's a little bit extra information that you're going to learn in third year, but just focus again on the mechanism and the adverse effects. All right, and good luck.